Hello, fellow conference attendee. My name is Shalaw Sallah from the University of Manchester, and our recent publication is entitled Using an Integrative Machine Learning Approach Utilizing Homology Modeling to Clinically Interpret Genetic Variants, CAC-9-1F as an exemplar. So I'd like to take a couple of minutes to introduce the gene, the disease, and the problem, as well as our proposed solution. So congenital stationary night blindness, type 2, or CSMB2, is a non-progressive retinal disorder. It's a rod-cone synaptic dysfunction. Its phenotypes include reduced visual acuity, nearsightedness, involuntary eye movement, and unaligned eyes. CAC-9-1F is one of 3 genes, mutations in which are associated with CSMB type 2. So CAC-9-1F is an X-linked gene. It encodes for a transmembrane voltage-gated calcium ion channel. This channel regulates the influx of calcium ion, resulting in continuous glutamate release from photoreceptor synapse in the dark to bipolar cells. 30% of variants are missense. Most of which are classed as variants of unknown significance. And that's essentially an inconclusive diagnosis. So this is shown in this lab report as an example from the Manchester lab, in which a CAC-9-1F missense variant has been flagged in a patient with phenotypes suggestive of CSMB. In the report, further down, we see that despite some evidence suggestive of it being causative, however, due to lack of sufficient evidence, as per the ISMB guidelines, the missense variant is regarded as a VUS. So the problem is experimental characterization of all VUS variants is impractical, and accurately predicting variants is difficult. So tools are often inconsistent and contradictory in their predictions. So these are prediction tools or in silico tools. Therefore, we propose using a gene-specific approach to protein variant interpretation through integrating genetic and protein structure data. To do this, we have pulled missense variants, CAC-9-1F variants, disease-associated as well as benign, putatively benign variants from different databases. We have modeled the protein structure, the structure of the protein, the ion channel, and we have used this model to differentiate between the disease-associated from the benign variants using the variant features such as conservation, physicochemical properties, variant clustering in the protein, as well as atomic overlaps, measuring atomic overlaps or van der Waals clashes. These analyzed features were then used to train a classifier model, a binary model. In the results section, we show that the protein model on the left, and on the right, atomic packing interactions between different atoms of residues or different residues in a protein. Then we show the results of the 3 evaluation metrics: the area under the receiver operating characteristic curve or ROC curve, and the area under the precision recall curve or the PR curve, as well as the Matthews correlation coefficient test or the MCC test. Using these, we've compared the prediction performance of the protein-specific classifier, which we have named PROSPER, short for Protein-Specific Variant Interpreter, versus the prediction of the other tools that we have considered in the study. The first graph, the true positive against false positive rates, or the ROC curve, Prosper matches the best tool. In the PR curve, or the precision recall curve on the other side, Prosper outperforms the other tools. And then further down in the table, the Matthews correlation coefficient test, or the MCC, results show Prosper to outperform the other tools by a large margin. The MCC test represents an overall correlation between the predictions of the tools and the true class of the variance. So we can conclude that analyzing CAGNA1F changes at the structural level is highly predictive of functional changes, which can then be used to improve variant interpretation. A protein-specific analysis facilitates a higher performance and more accurate prediction. This can be used as a standalone diagnostic tool to facilitate precise and timely diagnosis. We have replicated the performance of this approach in other genes and diseases, and we are developing models to interpret small in-frame indel variants. Thank you for listening.